Good. Alrighty, I think we are good to go.
All right. Hello, everyone. I'm Jamie Cate. I'm a professor of chemistry and molecular and cell biology at UC Berkeley, and where I've been for over a couple decades now. And I'm going to tell you about work that my lab has been doing as part of the Center for Genetically Encoded Materials, which is how I met Kate Adamala. So why don't I go ahead and share my screen, see if I can get this going. And is that working for everybody there as you see it, Evan?
Good, Jamie. Yep.
All right. So I'm going to see about whether I can hide this. Do you see this bar down here, or is it completely clear for you? Oh, you're
Sorry. We do not. We see your
okay.
arrow
So that's
over the
good.
CGEM
That's
logo.
fine. I will I'll maybe leave it up there just in case I see a question in the chat. So why don't I let me just see if I can go back here. It's not letting me go backwards. It only wants to go forward. Let me try this again. Sometimes Keynote does not play well with Zoom. Anyways, well, what I want to tell you about today is efforts in my lab and in CGEM, Center for Genetically Encoded Materials, to engineer the ribosome to synthesize new sequence-defined polymers. So what do I mean by sequence-defined polymers? We can think of proteins as sequence-defined polymers. They are encoded in messenger RNA, and then this, which has a 4-letter nucleic acid code, and then that is translated into a polymer by the ribosome, and uses, that's a 20-letter code that uses this acyl transfer chemistry to make the different covalent bonds. And we're using, nature uses 20 plus or minus a couple more natural alpha amino acids to do this, and which is represented by these R groups. And from those 20 natural amino acids, we get proteins with incredible diversity of shape and function, and all the diversity of life around us. But as chemists, when we think about these 20 natural amino acids, the diversity of these functional groups that come off here, where the R groups are actually not that diverse. There's hydrophobic, hydrophilic, some charge, some redox chemistry that's available. But chemists love to have a lot more diversity. And so in work that was pioneered by Peter Schultz at UC Berkeley, and now has been expanded incredibly, groups have figured out how to now incorporate non-natural side chains into proteins with dozens, if not hundreds, of different R groups now available. But one thing that's still common amongst all these is the backbone. And what we would like to be able to do is to change the backbone chemistry, which I've highlighted here in blue. And this could have changed the way the polymers fold and their stability and things like that. And I'm just showing examples of types of backbone chemistry that might be of interest: beta amino acids, cyclic beta amino acids, alpha hydroxy acids, malanates, things like that. The problem is right now, these non-natural backbones are very hard to incorporate into polymers using the ribosome. It's limited to very short peptides. It depends on sequence context, and multiple incorporations are incredibly difficult. Basically, it's been possible, with very few exceptions, to only incorporate one of these, a couple of these at a time, maybe. So this is where the Center for Genetically Encoded Materials comes in, and I have the website down here. And we're trying to engineer the translation system to try to improve this incorporation of non-natural backbones. So what I want to tell you about today is the part that my lab is playing. I've been studying the ribosome my entire independent career. And when we think about what needs to be done to incorporate these non-natural backbones, you have to have amino acyl tRNA synthetases and tRNAs. These need to be delivered to the ribosome, and then the ribosome itself has to be able to incorporate these into polymers but we need to do this in a way that's orthogonal from the rest of the cell, because the cell needs normal proteins to live and thrive. And so we've been working on aspects of orthogonality, the chemistry of how the ribosome incorporates these non-natural backbones. And then, as you will see, that's made us also start to think about the stability of the ribosome. So I'm going to tell you, I won't get into orthogonality today, but I'll talk about chemistry and stability. So let's start with chemistry. And what I want to emphasize here is that we can now actually use cryo-electron microscopy to really look at the chemistry of translation in detail. And what I'm showing here is a cryo-EM map for a structure of the bacterial ribosome that we determined a few years ago at 2 angstrom resolution. And this is just the Fourier shell correlation to show you that in some parts of the ribosome, like the large ribosomal subunit, we actually get better than 2 angstrom resolution. And what can we do with this? So what I'd say is we're no longer in the era of cryo-EM blobology. And I've actually used blobology in the title of a paper in the past, but can't really do that nowadays. So what can we see at 2 angstrom resolution, or even better? We can now really see the details of what's going on in a structure. And this is just some areas of density for the small subunit and the large subunit of the ribosome. And I just want to emphasize how we can really we can distinguish hydrogen bonding distances versus van der Waals interactions. And the distances are very clear, better defined stereochemistry. We can see solvation. We can identify the geometry around ions and other things like that. So we really can define what's going on at a chemical level now, which was not so easy to do before. So how we've been using this. I want to tell you about how we've been thinking about the chemistry of translation using the example of aminobenzoic acid monomers. So these aminobenzoic acid derivatives would incorporate an aromatic group in the backbone of a polymer. And what can this do for you? Well, it can actually change the geometry that these polymers, the stereochemistry, and these polymers also have really interesting stability. This is an example of one of our fire retardant lab coats that we have at Cal. These types of backbones would just be completely different kind of robust properties than normal alpha amino acids. And so there's been interest in trying to incorporate these into polymers, even into proteins, but this has been difficult. And so this is an example from the Suga lab using these 3 monomers, the meta, ortho, and this APY version with the nitrogen in the ring. And what one can see is in vitro, in a system that's highly optimized to get the most out of these systems, you can get a single incorporation, but it does not really come close to the yield of just using a normal alpha amino acid. So why is this? We wanted to understand our first question using the wild type ribosome is how do these substrates bind in the catalytic center, the peptidyltransferase center of the ribosome, and see if we could understand why they were not being incorporated efficiently. So how did we do this? So Chandrima Majumdar in my lab and Joshua Walker in the Shephard's lab optimized a system for attaching these aminobenzoic acid derivatives to the 3' end of tRNAs, and we're actually substituting the 3' hydroxyl with an amine group here to make an amide linkage instead of an ester linkage. And the reason for this, and this is actually work that was pioneered when Yuri Polikonov was in the Tom Stice's lab, is in doing structural biology, the ester linkages in the ribosome are just too labile and interfere with the ability to get structures. So we use these kinds of substrates for the structural biology. And what we then did is we used FMET, tRNA, FMET in the P site. So we had a normal amino acid there. And then we would bind an A site tRNA, the substrate, with the aminobenzoic acid derivative. And with these substrates, we're able to get to very high resolution. Again, in the region of the peptidyltransferase center, often better than 2 angstrom resolution. So what do the maps look like when we do this? So we can actually see, this is the terminal adenosine of the tRNA for each of the maps. And we can see the density for the incoming substrate quite clearly in all the maps. In fact, with the meta ABZ, we can even see that the substrate can adopt 2 different conformations. So this nitrogen here in blue could be positioned in two different ways. And we could see that from the map. So now just think a little bit more about these are docking into the ribosome, and we can see them clearly. How well are they docking? So the first thing to look at is how do these substrates bind in the A site? And what I'm showing on the top is a structure of a wild type amino acid, alpha amino acid, a structure that Zoe Watson determined, just to show you what a normal substrate would look like. And what we can see is that there's this base pair with the A loop, and it's conserved with these aminobenzoic acid derivatives. There's really no problem at this stage. So at least in terms of how the ribosome is binding, the tRNA part of the substrates seem to be going in correctly. In fact, there's even this what's still called A site cleft, where the side chain kind of packs between two nucleotides in the large subunit RNA, A2451 and C2452 in E. coli numbering. And this is also conserved with aminobenzoic acids. So, okay, so that's all good. Where do things start to go really wrong? So again, what I'm going to show here are two normal amino acids. And when you have a P site and A site substrate, the attacking nucleophile has to be able to approach the carbonyl carbon roughly 3 angstroms away in the ground state. This is what's generally observed with alpha amino acids. And this is, again, a roughly 2 angstrom structure that Zoe Watson determined. And it's consistent with structures from Yuri Polikanov's group. And what we can see is with all of the aminobenzoic acids, this distance is larger, which is potentially one of the reasons why the chemistry is not proceeding as well. In fact, with the meta ABZ, which was the least active in Suga's experiments, you can see the distances are really quite far. So the structures could immediately show us why the meta ABZs would not be good substrates. They just don't get positioned in the active site well enough for the nucleophile to approach the carbonyl carbon. But again, we're working at 2 angstrom resolution, and we can actually see things that are even more subtle than this. So what I want to do is I want to talk about what we know about the chemistry of peptide bond formation based on structural biology. And this starts with work from Martin Schmeng in the Stites lab, and Yuri Polikanov also contributed this. And what Martin observed is that there's an induced fit mechanism for the peptidyl transferase reaction. And basically what he found is that when the A site was empty, nothing bound there are 2 nucleotides in the large subunit RNA that kind of protect the P site substrate, push the U2585, kind of packs on top of the ester linkage and positions the carbonyl carbon-oxygen bond such that a nucleophile couldn't attack. But once a site substrate binds, U2506 rearranges and U2585 moves out of the way. That allows the ester linkage to rotate, and then a nucleophile can attack. And so this is kind of a zoom in on that kind of conformational change that occurs. And I just want to kind of mention that this color scheme, we came up with this before Barbie came out. And so just want to make sure that's on the record. So what happens with the ABZ monomers? So here is an example of, again, what one would see with normal amino acids that Zoe Watson had. Again, U2585 is moved out of the way, and a nucleophile could approach what would be the ester linkage. With the APY complex, however, U2585 is not moved out of the way. U2506 is moved a little bit, but not far enough. And again, the carbonyl carbon is not in a position where a nucleophile could attack easily. And so again, there's this position of 2585 and then the orientation of in this case, an amide linkage, but where we think the ester linkage would be. So one other thing that we noticed from these structures is that water molecules involved in peptide bond formation were not properly positioned. So this again is work coming, starting with Yuri Polikanov in 2014, where they used X-ray crystallography to identify that in the peptidyltransferase center, there are likely three waters. This is one model for how peptide bond formation works. There's three waters that are involved in various steps of proton transfer in the various steps here in terms of like deprotonating the attacking substrate, and then at the end, protonating the leaving group. So these three. And we actually, with Zoe Watson, were able to recapitulate this using cryo-EM. Again, this is just kind of a validation that the cryo-EM is reproducing what was published before. And again, that's interesting because this is slightly more physiological conditions when you use, you can get that with cryo-EM. And so we could see this with normal substrates. What about with the aminobenzoic acid derivatives? Turns out that we don't see all three of the waters. In fact, we only see evidence for water 3. Waters 1 and 2 are not there. And so again, what that means is that the attacking substrate, there's nothing there to help deprotonate. Really, there's only something there that could maybe protonate the leaving group. So why do these substrates even react? Well, one thought is that the pKa's of the exocyclic amines in these cases are dropped dramatically. And so that maybe compensates for the absence, at least of water 1, which might allow for these to react. And it might explain why API is slightly, this one here is slightly better than the ortho AVZ. So what do we learn from this initial set of structures? Again, being able to use very high resolution cryo-EM, we could see that for aminobenzoic acid monomers, there's poor orientation of the nucleophiles. The induced fit mechanism is not functioning properly, and the result is disruption of water positioning required for catalysis. And so this has led us to start thinking about new ways to add flexibility so that the nucleophile could be oriented correctly, maybe by adjusting the size or stereochemistry of the monomer that would allow the induced fit mechanism. And then this does open up the possibility of modulating pKa values for optimal activity. So that's maybe one silver lining to that cloud. So, but that was with wild type ribosomes. What about making mutations in the ribosome? So now I'm going to switch gears and talk about another parallel track we had going on in the lab based on experiments that started in Alana Shephard's group. And basically, this goes back a few years to 2016 when the Shephard's group published a mutant ribosome that can incorporate beta amino acids into proteins, at least one, and do this inside cells. And so how did this work? Well, they randomized a region of the peptidyltransferase center. Here's where the two substrates come in that I've been talking about, the P site and A site tRNAs. And what they did is they had 12 nucleotides changed in the peptidyltransferase center of the ribosome. Now, this is, for those who think about translation a lot, that's just kind of shocking, considering that the peptidyltransferase center is one of the most conserved sequences on the planet, but they were able to get this activity, increased activity with beta amino acid incorporation. And so we wanted to try to understand how these ribosomes worked. And unfortunately, what we found is when we purified the ribosomes, we could only get 50S subunits, the large subunit, instead of the intact 70S ribosome. And what we saw, again, using cryo-EM, is that whole regions of the 50S subunit near the peptidyltransferase center were disordered. And that's domain 5 in the 23S ribosomal RNA, and which I'm showing here. So this is the secondary structure of the large subunit RNA, domains 1, 2, 3, 4, and 5, and then 6. And if you zoom in on domain 5, this whole region that I boxed here, we could not see density for it, indicating that it was misfolded or disordered. And that's probably one of the reasons why we couldn't get stable 70S ribosomes in vitro or activity in vitro. So what's going on if we think about why would this be disordered? Well, it turns out that the mutations that in the peptidyltransferase center disrupt key base triples. When you look at the substitutions that were made, this 2063C to an A would be a problem. And then 2507 C to a U would also not kind of fit very well here. And so we thought that maybe one of the reasons that these ribosomes were struggling is because of tertiary structure being disrupted. Again, this notice the date here. So this came out again before the Barbie movie. So what did we learn from that work on this ribosome that can incorporate beta amino acids is that in addition to, even though these worked a little bit inside cells, they were not really going to be a great way to start trying to improve them because they introduced a major assembly defect as part of their increased activity with the beta amino acids. And so we wanted to start thinking about selecting for proper folding in addition to function. So how does that work? So now I'm going to switch gears, and this is again where we are now starting to think about ribosome stability based on what we learned about by making mutations in the peptidyltransferase center. So maybe where to start is where a directed evolution of proteins. So when groups try to evolve proteins, one of the things that they often will do is start with a thermostable scaffold. Basically start with a protein from a thermophile, and in making mutations that increase a particular activity, the protein may be destabilized thermodynamically, but that may be okay because it would move it into a temperature range where we may want to use it, say 37 degrees, instead of working at 65 degrees Celsius. So our thought was maybe we could take a similar approach with the ribosomes. So for example, if we wanted to be able to engineer ribosomes, perhaps we could stabilize the ribosomal RNA to get better engineering outcomes. So we just face a challenge, though, because the core of the ribosome, the peptidyltransferase center, is nearly universally conserved, but it turns out it's not entirely universally conserved. And so we went looking in the SILVA database, which is, as it says, a high-quality ribosomal RNA database, and used phylogenetics to try to find mutations that could stabilize the E. coli ribosome. And why the E. coli ribosome? Well, as probably most people on this call know, most of synthetic biology advances, they start in E. coli. A lot of the infrastructure for doing this is uses E. coli as a framework. So we want to be able to start with the E. coli ribosome and just identify mutations in the E. coli ribosome that would make it more stable. So I want to come back and kind of emphasize again where the problem is. The peptidyltransferase center was misfolded in the example of the P7A7 beta amino acid incorporating ribosome. But fortuitously for us, it also turns out that domain 5 is the last of the domains of the ribosome to fold, interestingly enough. So that's the part with the peptidyltransferase center. And this is work Christian Spahn and has done work on this, and Jamie Williamson, and basically showing that domain 5 is the last part of the ribosome that actually folds. So this problem here is kind of parallels that. The rest of the ribosome was able to fold, but not the domain 5. So we started looking in domain 5, and we wanted to focus, we first focused on the A loop. Again, this is the part of the ribosomal RNA that base pairs with the A site substrate, which I had shown before with aminobenzoic acid derivatives. And so we thought that maybe we could try to manipulate things in that region. So it turns out, so this G here is essentially universally conserved, but the neighboring U's are not universally conserved. They're mostly, but in a few archaea, they actually are change. So they change to C's. And what's interesting is we found that they turn out to be 2 C's in hyperthermophiles from the thermoprotei, which are kind of shown here on the right. And all the ones with the star kind of grouped together in the ones that grow at the highest temperature. It's not universal amongst those, but it is highly enriched. And so we thought we might explore mutations in the A loop to see if we could have an effect on E. coli ribosome stability. So I wouldn't be telling you about this if we didn't see something interesting happening. So maybe what I can do is explain what we're doing in this assay. Basically, we express the ribosome in E. coli with a tag, and it's actually a MS2 RNA hairpin that binds the MS2 protein. And then we can affinity purify the mutant 50S subunits and use them in vitro translation reactions. Now, it turns out that the way we originally used the MS2 tag, the way it had been used since 2005, in fact, actually destabilizes the 50S subunit. And we only could detect this because Amos Nisley in the lab developed this great assay where what he would do is purify the 50S subunits, heat them up separately from the rest of the translation machinery, cool them back down, and then put them in an in vitro translation reaction, say the pure reaction. And so what he found is that this tag was actually destabilizing. In a way, this is kind of like what we see with peptidyltransferase center mutations. But then this effect could be rescued by incorporating these mutations in the A loop, with the CC mutation nearly completely rescuing this stability defect. And we include a 30S subunit only control because we're not sure what might be carried along with other components of the reaction. So that's always a control we use. And so we also took these purified 50S subunits and did a heat after the heat treatment, just look to see what they would look like on a sucrose gradient fractionation. And so in the dashed lines are 50S subunits purified and then just incubated at a normal temperature, 37 degrees Celsius, and you get a very nice sharp peak on the gradient. But if we first do the heat treatment, what we can see is you lose a little bit of the wild type 50S, but this one with the MS2 tag really is destabilized. And as we add in the C mutations, we recover the folding of those subunits based on the sucrose gradient fractionation. So this is a start. We're think that we're making progress in terms of how we would maybe want to start with CC mutant ribosomes for doing directed evolution. But is there more that we might be able to do? So coming back to the P7A7 ribosomes, one of the other things that we had noticed in this study is that ribosomal proteins were sometimes depleted and or disordered in this structure. And so why is this interesting? Again, coming back to this Christian Spahn work, what one can see is that for a number of ribosomal proteins, they have these bits that kind of poke into the neighborhood of the peptidyltransferase center. And in fact, in their structural work, what they would see is that these parts in red were the last parts of these proteins to fold, just like the rest of the peptidyltransferase center. And this may be the most clear for UL4, UL3, and then UL2. It's not as clear with BL32, but basically the idea is that these parts of these proteins fold last. And so we thought maybe we could start thinking about the ribosomal proteins for ribosome stability. And so, in fact, when we go back and look at this P7A7 50S subunit, the map isn't fantastic, but one can see very similar patterns of disorder in UL2, UL3, and UL4 that were observed in this ribosome assembly pathway experiments from the Spahn lab. So again, coming back to the lessons from directed evolution, here we're thinking about maybe we could engineer ribosomes by first stabilizing ribosomal proteins before we go into the directed evolution. So how do we do this? So Amos and Nisley and Tamam Kamal in my lab came up with this plan where, based on the following insight, the core of the ribosome is highly conserved. We talked about the peptidyltransferase center in particular. And what's interesting is that our proteins from hyperthermophiles, which have to be stable in order to function at those temperatures, are interacting with the core of the ribosome, essentially close to nearly universally conserved bases. So even though they're in hyperthermophiles, they have to make contacts that they're basically constrained to contact the same kinds of ribosomal RNA as the RNA in E. coli. So when you look at these proteins, this is an example, what one sees is at the surface of the ribosome, the sequences are more divergent, and as they get closer to the PTC, they're more conserved. So the idea was to see if we can make a chimeric ribosomal protein that had E. coli sequences at the periphery and then moved to more hyperthermophilic sequences closer to the PTC. So I'm going to give you an example of ribosomal protein L4 to show you how this might work and the challenges in doing this. And the big challenge is that L4 turns out in bacteria, in E. coli, to control the transcription of the S10 operon. So another one of the ribosomal proteins. So in fact, all of these are ribosomal proteins. And so if UL4 levels are high, this represses all of the transcription for all of these ribosomal proteins. So there's this homeostasis for protein levels has to be very tightly regulated. And so, in fact, if we try to overexpress wild type E. coli UL4, what we see is that we get a lot of kind of misfolded kind of junk from those cells. And so overexpressing L4 is a problem. And in fact, if we look on an SDS gel, although we can see the tag UL4 incorporated into these particles, there are a lot of other proteins where the stoichiometry is not optimal. And so we decided not to work at the level of overexpression and shift our efforts to using CRISPR-Cas9 genome editing and just make the mutations directly, for example, in the operon itself at the chromosome level to try to maintain protein homeostasis. And so that was the idea that we wanted to pursue. And I just wanted to point out this particular system, the PEC-Cas and EC-gRNA that was published recently. These plasmids are available in AdGene, and Amos has been able to get these to work very well. So I'm just going to give you one example of a mutant we've just started to explore. This version 4.1 has 8 mutations in the protein, and it has a little bit of a lag phase, but it grows fairly well in E. coli. Again, this is the only version of L4 in these E. coli. And when purified, these subunits can work in vitro translation essentially as well as wild type ribosomes. But interestingly, again, if we use this approach of pre-incubating purified 50S subunits at high temperature, slow cooling, and then putting them into an in vitro translation reaction, this version, this chimeric UL4, does seem to give more activity. So increase the stability of these ribosomes to this heat treatment. So we think we're going to be able to use this kind of approach to build in additional stability into these ribosomes. So I've told you about how we're using phylogenetics at the RNA level, chimeric proteins. What other tools can we use? And the last part of my talk that I wanted to present today is efforts that we're trying in the lab to use deep learning. And this is inspired in part by AlphaFold, but really more by this second generation type of approach called ESM-Fold that is built on a large language model for proteins, and then has on top of that what is essentially the AlphaFold algorithm. And so what is the large language model? I thought I could use an analogy. If you think about large language models, one might think of things like GPT-2, GPT-3, or GPT-4. And what I want to do is distinguish those from things like ChatGPT. So ChatGPT is not these underlying large language models. It's basically the language models plus a whole bunch of additional layers, deep learning steps, lots of human intervention as well that occur on top of these self-learned kind of large language models. And so what we wanted to do initially was to see if we could develop a large language model for RNA instead of for protein. So I got inspired to try to do this by actually a GitHub repository put on the web by Andrej Karpathy. You may have heard of him he was the head of AI at Tesla for a while, and then he's now back at OpenAI. And he also just seems to really love to teach. So he has a bunch of videos that were helpful in getting an understanding of the code underlying deep learning models and some of the intuitions behind that code. And so I use this as a starting point to develop a large language model for RNA. And so in order to train a large language model, you need a training set. For OpenAI, it was the internet, and they've run into issues there with copyright infringement and things like that, maybe. But how do you do this for RNA? For protein, it was UniProt and the PDB, things like that. So what I did is I went back to the Silva database as a place to start. And because they've done a good job of trying to curate ribosomal RNA. And so thought I could start there. And so to do the initial model, I basically used the Silva database where I took the bacterial taxonomy and high quality sequences and alignments. And I kept E. coli to the side. I grabbed that sequence as well, but just for validation, I wanted to keep it to the side. And that gave me just under 90,000 23S ribosomal RNA sequences to train on. So then how do I get a thermophilic training set? The idea there that I had was to use the Tempura database. This is a great database. If you haven't looked at this, I would highly recommend you take a look at it. It's basically stores information on microbes and their optimal temperature, growth temperature. You can get the 16S ribosomal RNA sequences for the small subunit, but it doesn't have the 23S. So I had to go track those down separately from the NCBI database and other places like that. But I was able to ID bacteria with optimal growth above 60 degrees Celsius, find those 23S ribosomal RNA sequences and save those for fine-tuning the original large language model that was based on the Silva database. And this gave me about 100 ribosomal RNA sequences to work with. So how does this work? Basically, what I did is I used that kind of GPT-like model to train a large language model for ribosomal RNA. And then after having that model, I could fine tune it using thermophilic sequences from the Tempura database. And then the idea, again, we want to keep an E. coli ribosome as the ribosome we're working with. So I basically fed into this model, just kind of like you would do with a GPT, a starting query sequence that was E. coli nucleotides, and tried to identify thermophilic mutations that we could make in that E. coli sequence. So basically what that meant is that I started with sequence over here in domain 1, which is thousands of nucleotides away from where I want to find thermophilic mutations. And so gratifyingly, this large language model that I was able to develop, this is early days and kind of kicking the tires and testing it out. But one of the things that we were able to pull out of that model, it was that it was able to identify these as promising mutations, the ones we'd already tested that gave us more thermostable ribosomes. So this is something that we're really excited to keep pursuing to see if we can add that on top of using phylogenetics for the ribosomal RNA and the ribosomal proteins to see if we can use a deep learning approach for additional mutations. So with that, I just want to close, just kind of get back to the big picture of what really gets me excited about this project in CGen. So if you think about life on this planet, there are really only 3 polymers that have been subjected to evolution. One is, of course, proteins, and then RNA and DNA. I'm not actually showing a DNA example, but these are really the only 3 polymers that have been subjected to evolution. And so if we're able to change the backbone chemistry of polymers made by the ribosome, we might be in a position where we could evolve an entirely new kind of polymer, which I think would be really cool. With that, I will, that's again, where if we're successful in CGen, that's one of the things we'll be able to do. With that, I will just want to recognize people in the lab who've been doing the work. I talked about work from Amos, Chandrima, and let's see, also Zoe Watson and Fred Ward, both of whom have left the lab, along with Tamam and his work with Amos today. But then there are a number of other people in the lab who are participating in this, and this was a collaboration with the Shephards group and Ara Abramian at Schrodinger. I didn't talk in detail about that. And then also the Jill Banfield lab has been helpful on the phylogenetics. So with that, I will I'm going to stop sharing and we'll take any questions.
Alrighty, Jamie, thank you so much. Really fascinating work. I have a few questions, which I'll ask while people start hopefully typing them in chat or raising on the video. But so I was wondering about post-transcriptional modifications of the ribosome in the thermal files. There are generally more post-transcriptional modifications on our RNA. And are you considering, kind of matching either with a protein-based or RNA-based approaches, including considerations for post-transcriptional modifications?
Yeah, that's a great question, Evan. And we'd love to be able to do that. One of the problems with those is that they're not very well mapped across organisms. We'd love to have a more detailed understanding of where those are. And there's some implications for, 2' O-methyl groups often stabilize secondary structure and things like that. But our challenges, and so like the deep learning approach, we miss that. That's just a weakness that I'm not sure how we will solve. But there are still, one of the things that was interesting is we made that CC mutation. There is a modification to a neighboring nucleotide. It was completely fine. So that was still there. So it didn't interfere with that modification. So, but you're right. Those are things that I think require a bit more work on the experimental side to really just do a thorough job of mapping where those modifications are, especially in extremophiles. Yeah. So then Lynn was saying, well, why are polysaccharides or even lipids not subject to evolution? Well, I'm talking about in direct sequence space. They are not, right? The enzymes are, but the feedback is not at this so direct at the sequence level. So this is kind of what I mean by that. I hope that answers Lynn's question about what I mean by subjecting polymers to evolution. And then Maya asked, just curious why LLMs over something like diffusion models, which is more mutation-y in its training in theory. I agree. I am not a computer scientist by background, and so I thought I would start with large language models. And part of that is just they've been very successful in the textual sphere. Diffusion models, I think, have been more used in image and things like that, but it's possible to use them, I think, in the down the road. Yeah. Okay, and then have you considered directed evolution approach? So Michael Nguyen is asking, have you considered a directed evolution approach using a library generated by the LLM? That's definitely something that we're thinking about. How we were trying to decide how to get a large enough library that we could use. Again, these are big sequences. And so if we could focus in on domain 5, that might be of the ribosomal RNA, that might be a feasible approach. It's just a matter of scaling the throughput on the experimental side and the affordability on that side with how much we can get out of the large language model. So we're starting mainly with kind of just point mutations to see what happens.
All righty. Any other questions? All right. If not, I'd like to thank Jamie again and for you all for coming in today, and enjoy the rest of your Monday.
All right, Evan, thanks.
See you later.
Take care.